Hi everybody, welcome back to AP Biology. I'm Mr. Menike, and today we're going to talk a little bit about cellular communication as part of the human body lesson that we need to finish for our AP exam. Cellular communication is a way for cells to contact each other in order to maintain homeostasis. Your body has to be able to regulate all the cellular processes, your metabolism, your temperature, the rate of digestion, rate of circulation, breathing rate, all of this, including growth and reproduction, all of those life functions are regulated by cellular communication. The three systems that are involved with cellular communication most directly are the immune system, the nervous system, and the endocrine system. And so we'll be talking about those three systems separately. This is just going to serve as a little overview for you, and you can pause this at any time if you need to take a screen capture or you want to print something out, so feel free to do that. So the first thing we should talk a little bit about is what are the three kinds of cell signaling that occur. And the first thing that we should know is most, but not all, most chemical messengers are proteins. There are also chemical messengers that are, for example, steroids. Steroids would be, for example, sex hormones or corticosteroids that are involved in cellular communication, especially in the endocrine system. But proteins make up a large part of the messengers that regulate how your cells respond to a stimulus and then allow those cells to respond appropriately by either changing metabolic function, changing speed of their metabolism, protecting themselves, increasing the rate of transcription or translation expression of genes. So the first local signal that we should mention over a very short distance would be the immune system. Think about how the immune system actually controls and coordinates what we call self versus non-self. How your cells recognize what's you and what's not you, foreign invaders. And in order for them to do that, they have to literally shake hands. They shake proteins, but they shake hands. They literally contact each other in order to identify self versus non-self. And this idea of cell recognition would be like you shaking somebody else's hand. The other kind of cell communication that works over a short distance is the synapse or synaptic distance, and that is involved with the nervous system and specifically the proteins that are neurotransmitters. Think about the idea of being pretty close to somebody, and instead of shaking hands, you're passing the keys to them. You're giving a soft toss from one person to the next. That's kind of the way the nerve cells regulate their distance away from each other and still allow for communication to happen. There's a gap, there's a space, but still something is being conveyed between one cell and the other. It's a short distance, but it's not full contact between the two nerve cells. They don't do that. The last kind of cell communication is long distance signaling. And long distance signaling involves your hormonal system called the endocrine system. The endocrine system is very important because it's sort of like long tossing a football between two people, passing something downfield, a long distance, and still the message gets across. In order for that to happen in your body, this involves the circulatory system, because the circulatory system's job is to move material around through your body so that, for example, hormones can travel long distances between your brain and other organs in the body that need to be regulated. For example, your thyroid gland, or perhaps your adrenal glands, or perhaps the gonads. All of this involves some sort of communication that allows you to regulate your metabolism's speed and efficacy. Feedback, generally in the human body, is considered negative feedback if it modulates or regulates the signal so that the signal isn't overexpressed. We don't want too many hormones being expressed. And so the target cells that receive a message usually send a message back to the gland that's sending the message saying, we have enough hormones, let's regulate at a decent operating level, a metabolic level that maintains homeostasis. Positive feedback is sort of like giving birth or lactating, where when you have one signal, the signal is amplified and that signal continues to increase in intensity until you take the signal away. That's not homeostasis. Negative feedback is. We'll get into that a little bit more when we talk specifically about the endocrine system. But for today, we're just going to discuss in general terms how cells communicate with each other. We have an animal cell on the left side here, and we have plant cells on the right side. And animal cells have, if they're touching each other, they have little gaps in their cell membranes. And if you notice, take this picture and we kind of magnify what's happening in the cell membrane itself, there are these very special proteins here, these connexon proteins that can be either open or closed to allow material to pass through between one cell and the other cell. And so these are called gap junctions. And gap junctions are important for cells to coordinate with each other or to send materials between each other. Heart cells have gap junctions that allow them to coordinate the contraction speed so that the entire heart is contracting pretty much at the same rate, all of those muscle cells, those striated cardiac cells. On the plasmodesmata side of the plant, plasmodesmata are not proteins. They're literally gaps in the cell wall between two plant cells that are lined up pretty nicely to allow materials to pass through. And as you can see in the picture down here, we have the plasmodesmata that it's allowing materials to either pass through the cytoplasm in yellow or literally within the cell wall in green. So let me take a few minutes to go through each of the systems that I mentioned, the immune system, the nervous system, and the endocrine system, in a little bit more detail. Here we have an example of a local signal taking place between two immune system cells. And so immune system cell number 1 over here is communicating with immune system cell number 2 over there by the means of receptor proteins. And a lot of these proteins are called CD proteins, which help for cells to determine or differentiate between what we call self versus non-self. And if the cells are you, they're self. And if the cells are not you, a foreign invader, bacteria, for example, or a parasite, then that's non-self. If the proteins are not recognized, it could be viral, and your cells are going to try to mount a response against it. And so that response is here. These arrows represent some sort of change in the second cell. That change is usually some cascade of metabolic activity. And what that means is one set of reactions sets off another set of reactions that then tells the cell, well, maybe we should speed up metabolism, slow down metabolism, increase gene expression, lock the cell down, tell the cell to make more antibodies, depends on the situation. In a little bit more detailed picture of the cell, we have two white blood cells down here at the bottom. These are your immune system cells. And white blood cells, for example, a dendritic cell, is sort of like a phagocyte. Its job is to run around and eat things that don't belong. Whereas a T cell, they are regulatory cells. And regulatory cells, its job is to communicate with the rest of the immune system and coordinate an attack against something that's non-self. Over here we have a tumor cell, and even though a tumor cell used to be self, it's gone rogue, and it started to ignore the cell cycle and become non-self. And the way that your cells recognize all of this is they're looking at all of these protein receptors that you see here. And all of these protein receptors are what allow cells to both coordinate with each other and also recognize non-self versus self. Very important part of your immune system. You can also tell that all of these cells are touching each other. They're touching using their proteins. And so this is a very local signal. They're communicating by touching. That's the job of the immune system. It's pretty much like checking your badge, your ID badge, by literally taking your ID badge, holding it up, and reading it to make sure you are self and you belong. Because if you have the wrong ID badge or no ID badge, you don't belong and you must be removed. Another local signaling type of cell communication occurs in the nervous system. And in this particular picture, we have, again, two cells talking with each other. We have the sending cell, which is over here, and that's known as the pre-synapse cell or presynaptic cell. We have the synapse, which is the gap between the two cells in the middle, and then the receiving cell, the postsynapse cell. These little dendrites are what the cells use to receive stimulus from either the outside world or from each other. And then finally, the action potential that you see over here, well, that is the electrical current traveling down the nerve cell through the use of the sodium-potassium pumps, basically just salts moving in and out, causing a charge or an action potential. If we were to take this image right here and just blow that part of the picture up, and we multiply that by, I don't know, 1,000 times, we look at that more clearly. So I'm taking the synapse and I'm just looking at what's happening in the synapse. Here's the synapse now down here. This is my gap between the two cells. And in this synapse, we have a large space. The space is allowing for my presynaptic cell, the cell that is over here, and my postsynaptic cell, the cell that's over here. One is going to send and the other is going to receive. And so my presynaptic cell is going to send these little packages or vesicles of neurotransmitters. In other words, they're like tiny little vacuoles that have stored in them chemicals that will jump across this synapse. As they do, they're going to be moving from the first cell to the second cell. All those little blue dots. And what they're going to do is they're going to attach to these receptors that we have here on the second cell. That will tell the second cell to either fire its own impulse or not fire an impulse, to excite or inhibit the second cell. When the second cell is done using those neurotransmitters, it actually recycles those neurotransmitters back to the first cell. So these receptors are very important because these receptors capture the neurotransmitters, and that is called a ligand-gated ion channel. A ligand is something that's traveling between two cells that serves as a message. It's a gated ion channel because it opens and closes in order to catch or not catch those neurotransmitters. This is an example of tossing the keys to somebody from a short distance. Finally, the other kind of signaling or communication between cells is the endocrine system. And in the endocrine system, like we said, it's a long distance signaling system. And in animals, we have hormones that will transmit those long distance messages using your circulatory system. And so an endocrine cell or a glandular cell will release its hormones into the circulatory system. It'll travel long distances, and those hormones will then attach to the target cell, telling the target cell what to do, how to act, how to control its metabolism. As an aside, plants also use hormones. They use their own vascular system to do that, usually through their xylem or phloem. And then sometimes they can release chemicals into the air to act as hormones, like ethylene gas, which actually helps for plants to ripen their fruits. So, if we were to simplify the idea of how a hormone acts upon a target cell, we have down here our signal molecule that could be, for example, a hormone. And the hormone has to attach to a receptor. And so the first thing that has to happen is the idea of reception. Reception is when the receptor fits with the hormone on a target cell. The hormone comes from some gland or endocrine cell and is released into the circulatory system, where this receptor that's sitting on the cell membrane, and this is the cell membrane here of our target cell, catches that hormone. Once that hormone is caught, it sets off a series of chemical reactions inside of the cell, and that's called transduction. And so inside of the cell, we have relay molecules that signal some sort of metabolic cascade of events. And so here we have a cascade that's occurring. Well, once that cascade goes through the motions of enzymatic and energetic responses, you lead to the actual activation of the cell response. And that could be, for example, transcription and translation. Here's an example of something called a G protein coupled response. A G protein is a kind of receptor that kind of acts like a switch. It's called a G protein because it's a protein that's high in the ability to regulate guanine. Guanine is one of the four nucleotides in DNA, but it's also a component of GTP, which is a relative of ATP. And in this kind of response, there's energy that's being utilized by the cell. And so here we have an inactive G protein, and here we have a protein receptor that has no hormone attached to it. And if you notice in the second picture, we actually have a signaling molecule. Let's try that again. We have a signaling molecule. That's our hormone. The hormone has attached itself to the receptor. So this is our hormone, sometimes called a ligand. And it has now activated this G protein. And we know that the G protein is activated because here's our energy. This is again similar to ATP. This is our cascade of events that we were talking about before. Well, if you notice in the third picture over here, this little switch molecule, this G protein, again, this is our G protein, this is active. Our G protein is active, and it's active because it's been phosphorylated, because GTP has activated it, and it is now attached to and bound to some enzyme that's sitting in the membrane. Once the enzyme is activated, it sets off this chemical or cellular response, which could again be transcription or translation. When the hormone is removed from the receptor, it resets the entire situation again. And so all of these molecules become inactive again. The enzyme is inactive because the G protein is inactive and not sitting in its little receptor over here. The phosphate group has fallen off, which is a decrease in energy now for the reaction, and there's no hormone sitting in our receptor. And so your cells can toggle between these two situations of being active and inactive using energy and enzymes to start a chemical reaction. Finally, just to kind of show you the difference between how steroids work versus proteins work, and we'll get into this a little bit more later on when we talk about the endocrine system. Steroid hormones are known as first messenger hormones. A first messenger hormone is a chemical that can pass right through the cell membrane. Steroids can do this because steroids, if we remember, are lipids. And so a steroid, I'm having trouble writing there. Steroid is a lipid. Lipids are found in your cell membrane, and your cell membrane, your plasma membrane, is made of lipids. That word looks terrible. Let's try that again. And so if we have a hormone that's made of lipids and a membrane that's made of lipids, then what happens is the hormone can pass right through the cell membrane. That's what we see happening in step number 2. The testosterone in this example is our hormone. And so we have a few different examples of hormones over here that are steroids: testosterone, cholesterol, and estrogens. And if you notice, the shape of the molecule is similar in all three. These are non-polar molecules. And because your cell membrane is made of non-polar molecules, it can pass right through non-polar molecules. Like dissolves like. Well, here's what's interesting. Testosterone, for example, attaches to a receptor, but that receptor is in your actual cell cytoplasm. And what happens is that receptor enters the nucleus and bonds to genes inside of your nucleus on your DNA. Testosterone literally attaches to the DNA inside of a cell and tells the cell to transcribe some RNA so that the RNA can go to a ribosome and translate it into specific proteins. This is what builds muscle mass and builds hair and changes sperm production in males, for example, during and after puberty. Testosterone is a very powerful hormone because it directly influences your DNA, because it is lipid soluble, and it influences your DNA by targeting genes that control reproduction. Estrogen does a very similar thing for females when it comes to the sexual response and the menstrual cycle for females. When we compare steroid hormones, which are called first messengers, because if you look at it, a steroid hormone is the only message that really is involved here. It goes from outside the cell directly to the cytoplasm, right into the nucleus, and changes your DNA. First messenger hormones are one messenger. Compare that to what are called second messengers. Second messengers are proteins, and proteins like insulin down here, this insulin molecule is very big and very polar. And so this hormone cannot pass through the cell membrane. This hormone attaches to the actual cell membrane receptor here, and that sets off a cascade of events with the G protein attaching to an enzyme. And if we look at the name of this chemical, it ends in ACE, so it's an enzyme. And that sets off a cascade of events occurring inside of the cell to finally trigger a cellular response. This is very different from a first messenger, which directly affects your DNA. This is affecting enzymes inside of the cell that affect the endoplasmic reticulum's ability to release calcium, which then act as more messengers, the second messenger, which then cause various proteins to be activated and sets off a response that the calcium ions are regulating. And so while the insulin is a first messenger, a ligand, it doesn't enter the cell. We need more molecules inside the cell to act as our second messengers. Our second messengers are actually the calcium ions, as well as some of the other proteins that are activated that are acting like messengers. I hope that was helpful today. We're going to get into each of these systems in more detail. I hope that if you have any questions, you'll contact me, and I hope to hear from you soon. Have a great day. Without your scissors and my chromosome, cut me up and do it clear. CRISPR-Cas9.